So, it's always good when studying the topic to kind of go into the history. How did we get to where we're at now? What is immunity? So, immunity kind of comes from a term that means exempt, and that's kind of originated when, way back in the day, people noticed that when there was a plague and people got the plague, they were immune from catching it again, and they were great. They were the people you wanted taking care of the sick people the next time the plague came around. So people noticed that there was some type of exemption that we developed if you already got something. And they didn't understand the basis of this, but people started making these types of observations. 2,000 years ago, maybe more, who knows, but this is what we have recorded. So, some of the earliest inklings of immunology were some in China and Turkey. There was a practice called, I believe it was, name eludes me, but basically they would take an agent like smallpox, and they would take the pustule that would build up on the smallpox, and they would basically take that and cut people and put that dried pustule into the cut, or they would have people inhale kind of a aerosol made out of this, and that would. They found that, and I don't know who did that the first time or who came up with this idea, but they would find that those people might get the disease, but it wouldn't be as lethal, and they would have a better chance of surviving. It wasn't, it wasn't like our modern vaccine, and I find it interesting that, nowadays there's a lot of people who are afraid to get vaccinated, and the risk is so minute, whereas back then you get this treatment and you might die from it, but at least your chances were better than if you actually got the disease outright. So I guess they were willing to do that. In fact, I guess when a wife of an English ambassador actually saw this practice and did it to her children so that they would get smallpox. So that was kind of the first iteration of a vaccine. So all the way back in Roman times, they were talking about, they had the term immunity. In the 15th century, there's this variolation, which is where you cut the skin and put in that agent, which happened to be a virus. And it may have been the first kind of practice of using what's called an attenuated virus. And then Edward Jenner, he was one of the first guys who kind of said, he kind of made a huge breakthrough that he saw that milkmaids weren't getting smallpox, and they were milking these cows that had cowpox. And so he kind of found out that basically we can use cowpox, which isn't lethal to people, to vaccinate. And he didn't call it vaccine, so he didn't coin that phrase. He came up with this procedure to basically use variolation using cowpox pus or injecting it, and then that would basically give you immunity to smallpox. So he invented the first vaccine. Louis Pasteur later on, much later on, almost 100 years later, he began to basically experiment, and he developed vaccines for, I believe it was rabies, anthrax, and he coined the phrase vaccine, kind of a nod back to Edward Jenner. Vaccine comes from the root vaca or cow, or I don't know what the French version was, but basically vaccine means cow something. So it's so basically that's where that originates, and then in some of his vaccines he developed, or actually Some people claim somebody else developed, but he kind of stole the idea of an attenuated version. So he was able to, he took bacteria and attenuated them, got a weaker version, and he kind of stumbled onto this too because he was injecting rabbits with, I think it was a cholera, and then those, he had an old culture of it, so he went out partying, came back after a week, and had a culture that had been sitting there for a week. And he used that to inject rabbits. And he injected some rabbits and they didn't die like they normally did. And so, again, lazy, or too lazy to get more rabbits. The next time he did the same thing, but he came back with a fresh culture of his bacteria, and he injected, but he didn't have enough rabbits, so he injected some of the rabbits that survived the time before, and found that, they didn't die, even of this fresh, non-attenuated bacterial agent. And so there he kind of got the basis of that vaccine. So he did a lot of experiments. Some of it was, some of what he found, was accidental and was great, so serendipitous, but that's how most of our experiment, a lot of our huge findings come through serendipity. So, thanks to kind of this field, we now have vaccines. We've developed vaccines for things like smallpox, diphtheria, rubella, mumps, whooping cough, and we can see that what was the prevalence before, after vaccines. A lot of these we've eradicated. Some of them are coming back because, again, we're not immunizing at the same level that we were before, and actually there have been whooping cough outbreaks even here in Gainesville. So, you had a question? So, kind of highlights the importance of a lot of these findings and the impact that it's made. As we were learning more about the immune system, we were trying to develop ideas and theories about what was going on. There were lots of theories, lots of ideas, and as we were developing microscopes, people started learning about, they saw these white blood cells that were phagocytosing or swallowing pathogens whole. And so we got the idea that, maybe that's where this immunity is coming from. And we're developing, we have these kind of cell-based responses. So let's see, I wish I had a board closer. So cell versus humoral. So we developed some theories, started developing theories that this immunity was cell-mediated because people saw white blood cells engulfing things, and so they started to get this idea, But then trying to fine-tune what is the component in blood that helps to give immunity. Other groups, Behring, and Behring won the Nobel Prize for his work, found that there was some type of, something in blood, in the humoral portion, the non-cellular portion of blood, that could transfer immunity from one animal to the other. So he argued that, there's really no cellular component. It's a humoral or a non-cellular component that gives immunity. And so that argument kind of went back and forth for quite a while. And through that debate, we learned about T cells and B cells, and antibodies were discovered, and the protection, and basically we argued about it back and forth for quite a while, and it turned out that both were right. So this is one of those cases where there were two theories that were both right. There's a cellular component to immunology, to immunity, and there's also a humoral element to immunity. And in some cases they work in conjunction with each other. So we'll be learning about both of those, focusing in on both of those elements and learning about antibodies, learning about what cells are involved, T cells, lymphocytes. So basically, we've got B cells, which is a cellular component, but those B cells make antibodies. Those antibodies basically are shed off of the B cells and into the humoral portion, into the serum of the blood, and circulate. And so there's your humoral portion, but still there's cell-mediated immunity, which is this adaptive response as well, where you have T cells. And you've probably heard of killer T cells or cytotoxic T cells. These are T cells that will come and directly attack pathogens or cells that are infected with the pathogen. Also, T cells and B cells work together. You've probably heard of helper cells or helper T cells. Helper T cells will actually become activated or become stimulated. They'll bind to their target antigen, and we'll talk about what an antigen is. But basically, some foreign thing become activated, start secreting cytokines, and then alert the B cells that, hey, there's something off here. And then the B cells will start, if they become activated, will start to produce antibodies and shed antibodies. So they kind of work in conjunction with each other and alert each other to something that's bad. And I can say it's a pathogen, it could be anything, because realistically, your immune system doesn't know the difference between a bug and some foreign substance that has been put in there. It's just, its job is to kind of recognize something that's not supposed to be there, something that might harm you. So, then we start to develop, okay, we've got cells, we've got something in the blood. What is going on? Because how are we developing immunity to things? So there was this selective theory that Paul Ehrlich came up with, which is pretty amazing that he could come up with this theory without any molecular knowledge of what was going on, and really hit the nail on the head. He was right. He was dead on in his theories that there were these cells, and they had these side chains or receptors, and these receptors kind of work like a lock and key mechanism. He kind of took the idea from somebody who was studying enzymes and came up with that same type of lock and key mechanism, how a ligand or a receptor just perfectly fit that target, and then by doing so, it would then basically start to secrete those and then shed those off in the form of antibodies, and that would attack and bind to and target something for getting rid of. And so that was his theory. And his theory was that cells, there were these cells floating around that had lots of different receptors on them and lots of different lock and key combinations, and they were just kind of looking around, and if they found something that fit, then they would kind of start to make these antibodies. So that's the only place where he was off or wrong, was that cells, every individual cell doesn't have multiple receptors. It has one single receptor. We just have lots and lots and lots of cells with lots and lots of individual receptors. And when that one cell contacts its target antigen, then it will divide and start and mount an immune response. So the only place he was off was he thought one cell had multiple different receptors. The other theory, which was proven wrong, was the instructional theory that that cells have these receptors that would recognize and conform to the shape of something, and then they would kind of learn the shape of something and make a target for that thing. And so that was proven wrong. So I won't spend any more time on that one. So here's the actual drawing or representation of Paul Ehrlich's. So in immunology, that's one of those names that he was the first real immunologist, so you need to kind of know that name. This was his theory that you have these cells, they had different shaped receptors that matched different ligands or different antigens, and if that antigen matched up, it would start making more and more of these and start shedding them off, and that was his theory. And it was pretty much correct, except for that each cell only has one shape receptor. Okay, and we're going to talk about how is that possible? How do T cells and B cells develop with unique receptors and cover? I mean, your T cell, your normal average. So we have so many T cells circulating. We have the variability, I think it's 2 to the 7th potential combinations of T cell receptors. So I mean, it's a huge number of different T cell receptors you can have. So it's pretty impressive. And so we'll talk about how do you get that many different types of T cell receptors or B cell receptors or antibodies.
Can I ask you something?
Yes.
It's 2 times 5
Yeah.
for the order of the 7, right?
2.5 times 10 to the 7, sorry. So, basically, we, over time, understood that you have both the humoral response, driven majorly by B cells. Again, these B cells have these receptors, these antibodies or immunoglobulins that they used to call them. And they will, if they bind to an antigen, and so antigen, that's going to be your first kind of vocabulary term, other than cell versus humoral immunity. Antigens, basically anything that is foreign. It can be protein, it can be basically any type of molecule that's going to be recognized by your immune response can be an antigen. It can be RNA, it can be DNA, it can be sugars on the surface of bacteria. So glycoproteins, it could pretty much anything is an antigen. It's just a kind of catch-all term for some foreign thing that we're going to attack or recognize. So you've got B cells, they recognize an antigen, and they start to secrete antibodies. There's your humoral response. So if we took that blood now that's full of antibodies and spun it down, got all the cells down the bottom, sucked off the serum from the top, that serum would be full of antibodies. And we can use those antibodies to transfer immunity to people. And we still do that today. I think I got an immunoglobulin shot one time when I was going somewhere. And that's to help kind of, that's a practice that they actually started doing where they would get huge pools of serum and pull them together, get all the Igs or all the antibodies out of the serum, pull them together, and that would confer kind of immunity to somebody that was going to a place that they know had a lot of diseases that they may not have ever encountered. So that's the humoral element is also how a mother transfers immunity to her child, is through, she's not giving the child cells, but she's giving that child the antibodies through the milk that are going to help to also protect the child. And this is short-term protection. Whereas the cell-mediated response is going to be majorly through T cells. There are other cells, NK cells, but we're going to be focusing on the T cells because those are part of what we're going to call the adaptive immune response. So we're going to talk about innate versus adaptive responses in a sec. So you have cytotoxic T cells and helper T cells, and those T cells will recognize antigens being, and they recognize antigens being kind of presented to them. So we're going to talk about this in detail, but in kind of in a broad sense, almost all of the cells in our body have what are called Mhc molecules, major histocompatibility complex molecules that basically pick up all the antigens in the cell or in the surrounding area and kind of will present them. So they'll traffic through the cell to the cell surface, and they'll, like a little cup or a little two hands, they'll hold this antigen out and say, hey, this is what I'm all about, this is who I am. And then the T cells come around and just kind of, they're really promiscuous, they're checking everybody out until they find the person who has the antigen that might trigger their activation. And so you've got T cell receptors, which vary in shape, and then you have these Mhc molecules that are on almost all of our cells that have little chunks of peptides in them. They can have other things as well, but basically antigens. So they're presenting little chunks of antigens, and that kind of tells your immune system whether I'm self or whether I'm maybe infected with a virus. So you could have a virus come in and infect one of your own cells, and that's what's kind of depicted here. Viruses come in, and then you have these Mhc molecules that will pick up proteins, viral proteins, little peptides, will load them into their binding site that just kind of has a general loading area, and then they'll present them up. And then your T cell will come along and say, hey, wait up, you've got something, you're presenting something to me I haven't seen before, and I happen to have a receptor that binds to that, and so I'm going to mount a response. And here you can see now that here's where the link between our cellular immunity and humoral immunity comes. You get your T cells, your helper T cell, starts to secrete cytokines, and it activates your B cells and tells your B cells, hey, you should be alert to the presence of something foreign. There's something bad going on here. And then those can come in. It actually kind of brings in B cells, and the B cells start looking around, and if they see something if they see something that binds to their receptor, then they'll start to proliferate. So this is basically all of our adaptive immunity. Okay, so it's good to know why do we have an immune system? And so in the book, it kind of lumps everything into one of four groups because of viruses, which are very small and grow inside of our cells, and kind of that's why we've developed this interesting approach to where cells are going to always present what is being made in the cell to our T cells. So T cells are usually your first kind of line of defense for protecting against viruses. And that's why certain viruses that attack cells are pretty dangerous, like HIV, because HIV targets helper T cells. And if HIV kills enough helper T cells, then the cells that would actually recognize and help mount an immune response are gone. So there's so we have viruses, fungi. Some of these, are found everywhere. Candida Albicans is your basic yeast. It's basically everywhere. It's all over the place. And in most cases, because we have skin, because we have healthy immune systems, it's usually not a problem. It's usually more of a problem if you're immune suppressed, and we'll talk about how people are immune suppressed, types of immune suppression, whether that was a genetic defect, so primary type of suppression, or acquired, a secondary type. So we'll talk about that. But so fungi is much larger. Fungi are also tough because they're similar genetically to us. They're eukaryotes as well. Parasites are large. So we can deal with viruses or fungi through killing cells, which is sometimes if a cell gets infected with the virus, we'll sacrifice and kill that cell. But if, like a fungi, you can have individual fungi cells that are phagocytosed. Parasites are usually larger, so you can't. Our cells are much smaller. They're not going to probably phagocytose that parasite, but our cells can mount basically a damaging response to kill those cells, surround them, and create an environment in which those cells are going to die. And so, and that's how we would attack. So we've developed really mechanisms to attack all these bacteria. You can deal with bacteria through phagocytosis. They're much smaller than our cells. We can engulf those cells. We can tag those cells with antibodies, so antibodies will bind to the bacteria and then target them to be phagocytosed. So these are the different classes of kind of what for the most case, we deal with all of these things. Kind of our primary defense is skin. The acidity of our skin helps to prevent and control growth of these type of things. Mucous membranes help to slough off these things. And our immune system actually works together with those mucous membranes because whether we mount an immune response depends on the microenvironment. And depending on that environment, we may mount a different type of response. For example, I am breathing in antigens. I'm breathing in maybe bacteria. and that bacteria goes into my gut, into an acidic environment. It might kill that bacteria. It's going to line the mucous membranes. And so I have kind of this whole immune pathway where things that are coming into my body in that way, my immune cells are kind of looking for antigens to kind of protect against. And so we have kind of this interface between our blood and our intestines and our mucous membranes, where immune cells are really actively looking for things. But in the same vein, they're not attacking bacteria. So we have healthy, normal flora bacteria. We actually are tolerant to these bacteria because they're actually beneficial. So we develop kind of this agreement, I guess, with our immune system and bacteria. But then if those same bacteria were to get into our blood, we would attack them. So there's our immune responses may vary based on where they are encountering things. And then we'll talk about later on how there are actually immune privileged sites in the body as well. Central nervous system, so your brain. If any of you taken an anatomy, you know that there are immune cells, there are glial cells within the brain that have some immune function, but it's not the type of function that our immune cells in the rest of our body have. So we have to have kind of separate cells that won't damage our central nervous system as they mount a response. So we have some actual areas in our body that are called immune privileged sites. That would be like your brain, your eyes, your ovaries and testes, places where we're making sperm. So places where it's actually not beneficial to mount a massive immune response. So you don't want a lot of inflammation. So you don't want to be running away from a bear and trying to mount an immune response in your brain because it might shut things down. That inflammation might cause the brain to swell, might cause you to not be able to respond to the more immediate response of running from a bear. Same with sight. You don't want to be mounting an immune response and be blind for a week because you're trying to fight off an infection in your eye. So we've kind of developed sites in the body where immune responses are really suppressed.
So those are called the immune privileged sites?
Yep. So, and there's actually a lot of cool research being done where trying to understand how are they immune privileged sites? How do they shut down? And they're trying to create maybe immune privileged sites in like the pancreas or something if we want to try to turn off the immune response against insulin producing cells and diabetes or something like that. There are cancers of immune privileged sites that are really prolific and dangerous types of cancers. There's ocular melanoma. Is a melanoma that actually originates from the immune privileged site of the eye and is much more dangerous than melanoma. And the theory is because it originated from an immune privileged site, it is immune to the immune response that would recognize it as being bad, and therefore it is much more dangerous. And it really spreads to organs very quickly and is very lethal. So there's good and bad to this. So, and that's kind of, you're going to see that's a common theme in immunology, as we have. Immunology is a balance between protecting self and not damage self from so it's really a balance between here I have this system that's supposed to find things and kill them. And so I want to be ready for anything that could come up because viruses are always mutating, bacteria are always mutating. Who knows, I'm going to change regions and come into contact with something I've never seen before. So we have to have this system that can adapt to your environment. It can adapt to respond to anything, whether it's seen it or not. But in the same vein, I don't want my own immune system to melt my organs, to attack myself. And so there's this balance, and it's probably why over time our immune system kind of becomes a little bit, or the repertoire of things we're attacking. So T cells, they develop in the thymus. And the thymus is, when you're young, kicking out just this huge amount of T cells, and they have a lot of different T cell receptors. And as after puberty, that goes downhill, your thymus starts to atrophy, your repertoire becomes narrower, maybe you have a lot more T cells out there that already recognize something because we got sick when we were younger, and so they represent a larger percentage of what T cells we have. And so we may have evolved to kind of say, I'm going to protect you up until a certain point where you can mate, and after that it doesn't really matter if you die. But maybe I don't want to continue to produce lots of antigens because every time I'm producing new different T cell receptors, I have a chance that one of these might target you. So there's kind of this balance. It's really interesting. And that's where tumor immunology and autoimmune disease come in. Both of those are the fields of tumor immunology and autoimmune disease suppression are by kind of both sides of that. With autoimmune disease, the balance is tipped where you're attacking yourself. With tumor immunology, with tumors, your tumor derived from yourself, and we're trying to tip the balance to where we're going to attack self, but you want to be specific in that, and that's where the immune system is a great tool because it is very specific, much more specific than like chemotherapy. So immune responses rely on recognizing molecules. So we have a lot of what we're going to talk about in this class is going to be about what's called adaptive immunity. So that's going to be mounting responses to things, foreign things that we've never encountered before, mounting a specific response. But we also have what's called an innate immunity, and this is a more primitive, this developed much earlier, and these are specific responses that are in the germline. What I mean by that is the adaptive immunity is randomly generated. T cell receptors and B cell receptors they were made in various combinations and put out there. We select for ones that aren't going to respond to self, and then we have just this huge array of different T cells and B cell receptors that are part of the adaptive immune response. That will then, if they find something, will clonally expand and make it a response. Well, we have our innate immunity. We have all these cells, NK cells, different cells that are circulating in the body that have a receptor on them that is a specific receptor. And it has a broad range. It might be a receptor that recognizes glycoproteins that are common on bacterial cell walls. So you have, it's beneficial to have these floating around, and if they recognize something that is a common bacterial glycoprotein, it becomes activated. So these have these pattern recognition receptors, PRRs, that's what I put in red, because that's one you're going to need to know. Pattern recognition receptors, kind of a broad, they recognize a broad range of molecules that kind of will alert the body to, like the bacteria. These bind to pathogen-associated molecules, so PAMs. These are things that are associated with bacteria or virus or pathogens that are common. So it's not as specific, but it's reliable, it's always there. And there are some organisms, so there are fields of immunology that study sharks. Sharks have been around a really long time. There's a lot of immunologists in the lab. So I remember we were producing vaccines, whole cell vaccines to give to patients. And we had to find a way, we were producing them and then quickly giving them to patients before, so they were very viable. But we had to find a way to test to make sure that there weren't any pathogens in that mixture that we were going to inject into the bloodstream of the patient. You didn't want to be injecting bacteria. And so there were two ways of doing this. You could take a sample and culture it for, I think we had to culture it for 9 days. So that meant that when I made that vaccine, I I had to wait 9 days before I could give it to the patient. And so when you're making custom things for a patient, that's a huge amount of time. The other way to test for bacteria was actually use horseshoe crab blood. Now horseshoe crabs, they are old, really old. They've been around forever, and they have they have this incredible innate immunity. So they don't have this adaptive immunity. They just have receptors that have been around so long. They've developed receptors that pretty much recognize any pathogen. We haven't found a pathogen they don't recognize. And so you take horseshoe crab blood, you put it in, you mix it in, and if you get agglutination or you get this, if it becomes clotted, you say, oh, there's bacteria in there, I can't give this to the patient. It's instantaneous. So
That's when you asked me to say glycine, right?
Mhm.
Yep.
So that's something that we're using. That horseshoe crabs, they got it made. They've got a great, strong immune system. But we do have this adaptive immune system, which is really incredible. So other important concepts to understand: humoral and cell-mediated immunity rely on these surface receptors, and we're going to be learning about those and how they work, how we have this wide array of them. We're going to talk about and learn about how they are randomly generated, and these are going to be some of the more beefy, complex chapters. So I think we're going to have just 3 chapters in the exam where we're covering this. And the majority of these random receptor combinations that we made are actually deleted because either they recognize self or they don't have a high enough. So in the case of T cells, we have what's called, and we'll talk about this in later chapters, we have positive and negative selection in the thymus. This is where T cells, if they bind, so our thymus is really cool. It kind of, the theory is that it presents at low levels a little bit of everything. So it's kind of like the garbage burrito of the immune system or whatever. It has everything in it, and it presents everything. And as your T cells are developing, if they have a receptor that binds to those thymocytes, they are deleted, they are killed. Because you don't want T cells floating around that recognize you. And then, so in the most part, all the different combinations we produce either don't respond to our cell, and they do respond to our Mhc molecules, because those are the molecules that are presenting the antigen. So they have to kind of have a little bit of recognition for those. So that's what's called positive selection. And once they've gone through this process of selection, they're kicked out into your system, and they circulate and look for pathogens. But they still have this huge, broad array of complexity and randomness. Same for B cells. B cells, you can actually get even more than 2.5 times 10 to the 7th variations because you have hypermutation that happens later on. We'll talk about that. So you can have even a broader range of potential receptors that recognize different things. Then what happens, so another term you need to understand is the term clonal selection. And so here's where if a B cell or T cell recognizes something, if it binds to that antigen, it just randomly comes across something that it binds to, it is going to go through, it is going to start proliferating, and it is going to start dividing, and it's going to start kicking, and if it's a B cell, it's going to start, depending on the signals it gets, it might start kicking out antibody and releasing that into the bloodstream. So this is how and then eventually these cells not only are going to divide, but they're going to differentiate, and you're going to get a small population of memory cells, you're going to have effector cells that are going to attack things. So you're going to not only mount a response and kind of make a little army, but you're also going to have some sentinels that stay behind after you've mounted your immune response, after you've gotten rid of that disease, and you're going to have memory. And so memory is one of the key things in the adaptive immune response that you'll have. Yeah.
Is this going to be online?
Yep. I have this in Canvas. All the PowerPoints are in Canvas. So if you go on Canvas and go to modules, it's listed under lectures. Okay. So when a B cell or T cell interacts with its specific antigen, it's selected, becomes activated. As I said, the activation results in proliferation, producing a large number of clones. So this is this clonal selection. So kind of, we have stem cells. So this is just in a nutshell. You have a stem cell, it's in your bone marrow. If it's a B cell, it's going to develop in the bone marrow. It's going to make lots of different B cells, with lots of different receptors, lots of different antibodies. Those are going to have some type of selection. It's going to get rid of those that recognize us so that we're not killing ourselves. And those are going to go out into the bloodstream or into our lymph, and those are going to circulate and look for antigens. Number two, our lucky number two, found an antigen. It's a viral antigen. He binds to it. He gets tickled. It tickles that receptor. That receptor signals into the cell, hey, I found something. The cell then starts to proliferate. Now it's a little more complex than that, depending on its microenvironment, depending on where you are in development, depending on, do I have signals from cells? Are cells dying around me? Are cells happy around me? Is there helper T cells that are also activated? So it's going to, depending on the other signals it's getting, it's going to decide, am I going to proliferate? And so in this case, it is proliferating and making lots and lots of these that are now going to flood the area, circulate throughout the body, and look for more of this antigen, this pathogen, this virus. And so that's your kind of your clonal selection and expansion. So other concepts, we developed tolerance. Tolerance ensures that the immune system avoids destroying our own cells. That tolerance can come in, and like I said, when we develop our T cells or B cells, we get this selection, this negative selection that gets rid of candidates that probably are going to recognize ourselves. So that's how we develop tolerance. Early researchers also found that if they injected an antigen into baby mice, let's say, those baby mice later in life would be tolerant to that thing. They would not mount an immune response against it. So you're kind of you're educating the immune system early on that this is part of me, and they were now tolerant and would not attack that thing. Tolerance can happen in that initial period of making T cells or B cells, but it can also happen later on. There's new research that is focused on low-level chronic inflammation can lead to tolerance. And probably because if anything gets out that is going to react to self, we maybe don't want it. We want a second line of defense against things that are going to attack ourselves. So when they're out in the field, they have to look in context. Oh, I got tickled. I see something. I'm looking for danger signals. If there are no danger signals, then maybe this thing isn't. Maybe this thing is self, and I slipped through. So I'm gonna turn. Not only am I not going to attack it, but maybe I'll turn it into a suppressive cell. And there are these cells called T regulatory cells, and these are helper T cells that are actually suppressive to immune responses. And if you get enough of them in an area, they'll actually suppress immune responses in that area. And so that's actually what we were doing. We were trying to make regulatory T cells that would recognize antigens that were specific for insulin. And then we were trying to basically use those to suppress immune responses against insulin. And so that was a tool trying to, as we're learning more about how tolerance is built up, we were trying to use that to induce tolerance in autoimmune disease.
So the suppressive T cells are more like an anti-complex or anti-complement?
They secrete cytokines that basically will suppress that immune response. They have a couple different mechanisms to suppress the immune response, but they kind of create a microenvironment that is suppressive, not excitatory.
There's basically a don't worry about a response.
Yeah, and one of their mechanisms too is they act as what people call cytokine sink. And so if there's enough of them around, they kind of eat up all the cytokines that are inflammatory cytokines, and so they kind of calm things down in the area. And they also naturally helper cells will actually kind of go differentiate towards that to kind of, so that our immune response, when we're mounting a crazy immune response, you really want to shut it down after a while because you don't want it going off
How
for
do
a
you
while.
do that?
The cells themselves have basically a timer. A lot of cells have it's a timer that basically says, I want, I'm going to respond gangbusters for a couple, a day, a couple hours, and then they start secreting suppressive receptors, and they kind of they kind of together will shut down the immune response.
Do you inject some cytokine inhibitor into that local?
Some people have tried those, or they've tried genetically modifying cells to secrete those to be more continual. They've even produced
Less cytokine?
similar to insulin pumps that will pump in cytokine to mount or shut down the response. So yeah, as you understand what cytokines do, what cytokines are inhibitory, which ones are excitatory, you can kind of use that to your advantage. Other important concepts. Let's see how we're doing on this one. We've got about 10 more minutes. Okay, we should get through. So, important concept. Other is the response to pathogens. We have our innate, and I talked a little bit about this. We have our innate immunity and adaptive immunity. So the innate response time, minutes to hours, because you already have a lot of cells that have a lot of these receptors. You don't have to have this clonal expansion occur before you mount the response, which is why if I cut myself and I get some bacteria in my cut, within an hour that cut is puffy. I have inflammation. I have innate cells that recognize that bacteria. They have receptors for the sugar on the surface of that bacteria, and boom, minutes to hours, that whole area is red, angry, because I've mounted an innate immune response. I catch a cold. My kids went to school. They came home. They gave me a cold. I didn't start showing signs until 6 days later. Okay, that's because I mounted an adaptive. I've got fever now, get shakes, sweating, because I'm mounting this really massive adaptive response. And so these are how you can tell if something's innate or adaptive. Specificity, limited and fixed. You get what you get with the innate system. Either you recognize it or you don't, and it's a pretty broad range of things you recognize. Whereas adaptive, it's very diverse. There's a huge array of different things we can recognize, and it can actually adapt to improve, and we'll talk about how B cells do that over the course of the immune response. Responses to repeat infections. With innate immunity, those cells that recognize that are still there. They're still going to recognize it in the same way. Whereas adaptive, so if you have T cells or B cells that recognize something, they're going to leave those sentinels behind. They're going to have that memory. So now you have memory. So the next time you see that thing, the response is going to be much more rapid because I have more numbers of those cells already hanging out. Those cells are going to mount an even stronger response. They're going to secrete even more cytokines. Those cytokines, which are just molecules that, I guess I didn't define cytokines for you guys. Cytokines are kind of like hormones or chemokines in that they're secreted from these immune cells, and they kind of will create this, they will create a concentrated area that is stimulatory, where these cytokines can bind to other immune cells. And those cells then will start secreting chemokines. Those chemokines will bring in more cells, immune cells, and that's where that inflammation occurs and the puffiness occurs, because we're secreting all these chemokines and cytokines that are bringing more cells in and activating more cells. So the second response for the adaptive immune system is much stronger and much more quick. Major components. Well, part of the innate immune system are the barriers: skin, mucous membranes, phagocytes. Again, anybody who took anatomy will know the layers of the skin. Within those layers of the skin are a specialized immune cell, plasmacytoid dendritic cells. They're just sitting there, sentinels. And so they are ready to phagocytose any bacteria that make it through the top layers of the skin and alert immune systems too. That's where that inflammation comes in. So we have kind of these sentinel phagocytes sitting at all the barriers. They're kind of the guards. And then we have pattern recognition molecules that complement and that float around and will recognize certain pathogens that we've, through evolution, have encountered many, many, many times. And so that's, and then for the adaptive response, it's the majorly the T and B cells that are going to respond. Okay, so innate immune response is your first line of defense, fast but not specific. And actually, in some cases, damaging. So there's a local company called RTI they do research into transplants and healing. And so the immune system actually, when you have a cut or you have a wound, will actually create an environment that's really not good for healing. But it's also great for getting rid of bacteria and other potential pathogens. But if you do things in an aseptic way, you might want to suppress that immune response so that you can get healing, because it's not specific. This first line of defense, neutrophils, a lot of these phagocytic cells will come in, and they'll just create this damaging environment. So even though it's really important, it's not as specific and fine-tuned. Again, these use germline encoded recognition molecules that we've evolved to just have, and do not alter through time. And these are mostly used phagocytic cells. Our adaptive immune response, which includes humoral and cell-mediated responses, so antibodies and T cells using B cells that produce antibodies, T cells that are your cytotoxic cells and helper cells. It's slower to develop, 5 to 6 days, but uses randomly generated. It's very specific, and which is why a lot of people are trying to harness the immune system to attack tumor cells. So innate and adaptive immunity, they work together. So that innate immunity will also, those cells that recognize something, they'll also secrete cytokines. They create an inflammatory environment. They secrete chemokines that will bring in those adaptive cells. So they're the first alert, and then they bring in the adaptive immunity. So they usually work in conjunction. They stimulate and direct the adaptive response, basically. So here's kind of a graphical representation of what I explained. So here the light blue would be your innate response. It would respond very quickly to antigen A, and then if you reintroduce that antigen A later on, the response would be the same. So the magnitude of the immune response is going to be identical. Whereas, let's say we have antigen A, we have an innate response, it helps bring in adaptive cells, B cells, and T cells, and they take a while to expand and to become activated and to differentiate and really get rid of that infection. But once they have, then they go away and they create memory. And if you mount a secondary response later, the response is much greater. And this is great, except if it's against something that you normally come into contact, like an allergen. Let's say peanuts. So I have this initial response to peanuts. The next time I see peanuts, it's even larger, and this is where you can actually, this is where you tip the balance, and now you can actually have what's called anaphylaxis, which is you've mounted such a huge immune response that you're going to kill yourself. You basically you're secreting so many cytokines. You're actually, and a lot of these cytokines will make thing makes cell barriers more, they'll open them up, allow more cells to come in, and that's what happens. You'll get capillaries in the lungs that just start to become leaky, and you can actually asphyxiate, you can drown because of this anaphylactic response. So that's the dark side of immunity. So here's just a figure example, which I'm not going to spend a lot of time on. You have, again, like I explained, you have a pathogen that comes in. It is recognized by maybe an innate phagocytic cell. That cell then will traffic to the lymph nodes. It'll carry with it that antigen, and it will start to basically present that antigen. So you get these professional antigen presenting cells. A lot of the phagocytic cells in the body, what they do is they pick things up. They'll traffic to the lymph node. That's why when you get sick, you get those swollen lymph nodes, because all these cells are going to the lymph node. They've run to the lymph node. They're presenting antigens. So again, those Mhc molecules are presenting antigens, and the T cells and B cells are recognizing them and starting to proliferate. You've created this epicenter of proliferation, and then those T and B cells start to secrete cytokines, bringing more cells in. That lymph node is growing and getting painful, and then those cells will mount. They will proliferate and then mount a response to that, and B cells will start secreting antibodies, and T cells will start to proliferate and attack different cells, or the antigen or infected cells. So dysfunctions in the immune. Again, there's two. The good is what we've been talking about. The bad is and ugly are the immune system does things that you don't want it to do. So overly active, misdirected immune responses. Again, the immune system, doesn't have, these cells don't have brains. I'm not saying, oh, this guy's a pathogen, but this is just an allergen. So we don't know that, oh, this is pollen, this pollen's not going to hurt me. We don't know that. It's foreign. We recognize it. So we mount an immune response. We start secreting antibodies. That's why histamines are supposed to help to suppress that antibody production, those histones. And so antihistamines are going to help kind of reduce this. So we're actually now taking drugs trying to suppress immune responses against things that we encounter every day that aren't really harmful. So we'll talk about allergies and kind of how what allergies are and how we're trying to control them. Immunodeficiency. Primary genetic immunodeficiency is you're born without a certain gene that is really important for an immune response. And you can have there's different types. There's all the way up to skin, which is, this is where you have to live in a bubble because you just have no immune system. Or it could be something where you have most of the components of your immune system work, but maybe one aspect doesn't. So you're at a higher risk for infections or cancer. And that's kind of how we started learning that the immune system actually does alert your body and does attack cancer cells, because we found that people with certain immunodeficiencies had a higher rate of certain cancers. And we had found that mice that we bred with immunodeficiencies had a higher incidence of cancers. And so we started to learn that relationship between cancer and the immune system. We'll talk about that later. Secondary are immune suppression or immunodeficiencies that are acquired that aren't part of the germline. These are loss of immune function due to chemicals, due to HIV, where you're basically losing all your helper T cells. Transplants. We did not evolve to say, hey, it would be advantageous if I could just get an organ from anybody. We evolved to basically keep out anything that's foreign, but that now becomes a difficulty because foreign could be somebody else. We've developed, our immune systems not only has a broad range of T cell receptors and cells, but we have a huge array of what are called different alleles of different Mhc molecules. So those molecules that tell the immune system who are my cells, what are my cells. Those Mhc molecules, those recognition molecules, are different throughout populations. And because they're different, my immune cells are going to attack them. And so now that's bad because if I want an organ, I either need to find a really close match, genetically speaking, or I have to have something that doesn't have any of those markers at all on them, or I have to populate them with specific markers. And so some of the ways we get around that is if you get an organ, we have to immune suppress you so you don't attack that organ. Or you have to get an organ from a very close relative that's going to be a very close match. But even within relatives, very close relatives, there's variations in MHC, which is, it's great for making sure that somebody survives that plague, but it's not so good when I want to donate an organ. The other thing we're looking at is getting organs from animals, where we can produce animals that have those human markers that are specific to specific people, or using people's own cells to make new organs, which is really probably the easiest and most efficient way to deal with this, because you would get self, and that would not be recognized by your immune system. And then cancer again. We'll talk about in the cancer chapter how basically we all are getting cancer constantly. We have cells that mutate and start proliferating out of control. Our immune system, it sees things that are that are not normally expressed, and so it actually will pick up and kill those cells. You get cancer when you no longer recognize that that cancer has developed the ability to suppress the immune response. And so that's a common thing is cancer cells will create a very suppressive environment. We'll talk about that. Immunity is a complex subject. We're going to try to break it down into different layers and areas, which is we're going to go through all the chapters. But understanding immunology is going to allow us to really exploit it through vaccinations, which we've already done, through shutting down the autoimmune disease or ramping up cancer responses, which is really promising field. I have seen people with head and neck cancers that are just horrendous, where they have been able to treat those with immune responses, where they've taken T cells out of that patient. They put in a receptor that's specific for that cancer, and they basically melted that cancer away. I mean, it's amazing what they can do. And so, and those patients, other than having flu-like symptoms, they're not having other cell damage. They're not losing their hair. They're not having the effects that chemotherapy would have. So it's really understanding immunology will allow us to. Diabetes also affects so many people in over a long period of time. If we can harness the immune system to suppress that and to not attack self, then we would be able to help those people without suppressing their immune system.
May I ask you something?
Yeah.
You see the diabetes, you mean type 1?
Type 1, yes.
Yeah.
So there's an autoimmune type 1, and then there's the non-autoimmune type 2. It's more associated with obesity. And type 2 is the more common type, but you can have both.
Even people, normal weight, they still can have type
Yes.
2. Even skinny people.
Yeah. Oh, yeah. Okay, so, and for providing safer organ and tissue transplant, understanding immunology, understanding what is being recognized, that people are starting to produce pig organs that have human Mhc molecules on them that can be used in transplantation that will not be, that will be better tolerated. And the cool thing, I think, well, for me, the cool thing about immunology is we are still learning. This is what, I mean, you could probably go into any field where we're still learning things. We don't know everything. But immunology, really, we are still in our infancy in immunology, understanding. There's still so much that we don't understand. There's so much we do understand, but we are still making advances. We're still learning how to harness the immune system and what's so great about the immune system is you already have this mechanism in the body that is going around, looking through the body, that will get rid of bad cells, that will take care of good cells, that is already in place. If we just learn to harness that, we can actually use it to treat so many diseases in a much more specific manner. So that's where we're going to end today.